Hello, my name is Jordan. This is my very first time presenting a poster, so I decided to make a little video to practice my presentation and for the convenience of those who didn't get to catch me standing at my poster in person. I'm a brand new PhD student and a member of the Vecchiarelli Lab at the University of Michigan. This is me right over here. I'm really excited to show you guys some preliminary data on the aggregation and distribution of carboxysomes, which will be the focus of my project going forward. So without further ado, I'll jump right in. Bacteria do have organelles. My lab studies a protein-based organelle found in chemoautotrophic proteobacteria and cyanobacteria called the carboxysome. If you're not already familiar with carboxysomes, I'll go ahead and give you the rundown. Carboxysomes are a specific type of bacterial microcompartment. They're icosahedral, 20-sided, protein-based organelles that serve the cell by enhancing metabolic carbon fixation. How it achieves this is the carboxysome shell proteins are selectively permeable and they function to specifically encapsulate and enrich the carboxysome lumen with desired compounds like carbonic anhydrase and rubisco, which in turn attracts CO2 and lends itself to enhanced and effective fixation of atmospheric carbon into sugars for biomass production. What I want you to take away from this is carboxysomes function to encapsulate and concentrate enzymatic reactions, thereby increasing reaction efficiency and product yields. Carboxysomes are of great scientific interest due to their environmental implications. They're responsible for roughly half of global tooth fixation and generally enhancing our understanding of bacterial microcompartment organization. Looking at carboxysomes specifically will provide a basis for their minimal autonomous positioning systems to be used as regulators for a wide variety of desired cargoes, ideally for synthetic biology and biotech applications. And speaking of positioning systems, our lab recently identified the maintenance of carboxysome distribution system, which I'll refer to as the MCD system. It's composed of two partner proteins that work together to modulate carboxysome organization in bacteria. Protein 1, called MCDA in purple here, is an ATPase which non-specifically binds the bacterial nucleoid and forms dynamic oscillations on the nucleoid in response to its partner protein, MCDB. MCDB is an adapter protein that chases the MCDA gradient across the cell and specifically associates with carboxysomes, effectively connecting the carboxysome cargo to its positioning system. The oscillations direct carboxysome motion and equidistant positioning in the cell to optimize carboxysome functionality and ensure their inheritance after cell division. So we wanted to know what happens in the absence of MCD components. Here we show that in the absence of these proteins, carboxysomes will no longer be dynamically positioned along the nucleoid. Instead, carboxysomes are excluded from the nucleoid and form aggregates at the cell poles. I want to be clear in saying that the carboxysomes here at the pole are not amorphous clumps of unstructured protein. They're fully assembled carboxysomes that have essentially snapped or stacked together. Of course, this begs the question, will assembled carboxysomes that have been nucleate excluded at the poles get redistributed upon the reintroduction of our positioning system? So yeah, that was one of the questions we had in mind. Is the MCD system robust enough in function to redistribute a conglomerate of carboxysomes? Like for example, with these MCDA deletion cells. We observed there was a carboxysome aggregate formed at the pole, and we see that in our cells as well. And then we reintroduced our MCDA-B components through inducible expression. And after induction, we observed that the aggregation, the carboxysome intensity was distributed upon the nucleoid, which suggests that carboxysome aggregation is dynamically reversible. So I'll just walk you through this chymograph here. So we have three examples of like 3 cells over a time course. We see that to begin with, there are aggregates of carboxysomes at their pole. We see them get pulled onto the nucleoid and equally distributed over time. And let's see here. And after removing the inductant, we see this opposite effect. So we see that they're distributed, and then we see that they eventually aggregate again. So we see this opposite effect. We see that distributed carboxysomes eventually will snap back together, but still hang around the nucleoid. They still hang around the nucleoid enough, which suggests that there's still some amount of MCDA-B around in the cell, preventing the carboxysomes from fully falling off of the nucleoid, but not enough MCDAB to effectively distribute the carboxysome and prevent the aggregation to begin with. So our results here are really striking, and they demonstrate how the dynamic positioning of carboxysomes across the nucleoid is entirely dependent on the presence of both MCD components. So it's apparent that the MCD system is really clearly linked to dynamic carboxysome positioning. However, going forward, we really want to parse out MCDAB's mechanism of association during de novo carboxysome assembly. It's currently thought that the life cycle of a carboxysome is as follows. Carboxysomes assemble at the pole, they bud off of this procarboxysome, they mature and get pulled onto the nucleoid and get distributed, and eventually when dying, they're shuttled back to the poles to be disassembled, and their components are recycled to form new carboxysomes. This AB system is solely responsible for the experimental evidence we see and is a really strong connecting factor between these stages of the carboxysome life cycle. As for my future directions, I mentioned earlier why we care about researching carboxysome dynamics, and that's due to synthetic biology applications. I'm aiming to advance an interdisciplinary understanding of molecular delivery vehicles as a means to produce important compounds on an industrial scale. Additionally, this effect has only been observed in rod-shaped bacteria and cyanobacteria. Specifically, I aim to address how physiological and morphological differentiation in cyanobacteria influence carboxysome spatial regulation and distribution. And you can see there are a number of spherical and filamentous cell types, and I could go on and on about specialized cell differentiation within a filament and how I plan to observe carboxysome dynamics within these cells to deepen our knowledge. So please, if you see me, please ask me questions on this. I have a lot to say. And with that, I'll just say thank you guys so much for watching. If you see me at the conference, please feel free to stop me and we can have a conversation about these things. I can show you cool videos, or we can talk about whatever you want. If you have any ideas, comments, questions, concerns, etc. And if you don't see me at the conference, please just feel free to shoot me an email here and let me know what's going on. Again, thank you so much for watching, and I hope you enjoyed this video as much as I enjoyed making it. Thank you.